think a lot of people, when they think about animals in research, they often think about mice or primates, right? And these things are popular models to study in research because they're evolutionarily very similar to humans. But a lot of people don't think about things like zebrafish in research, and I think it's because they look very different. But in reality, you know, they have a lot of similar anatomy. They have eyes, they have a mouth, they have very similar skin to humans, for example. They have hearts, they have all these components that we still use as humans. And interestingly, they actually have about 70% similarities in their genomes compared to human genomes. And this is actually one of the most interesting parts about zebrafish too, because we've developed a whole set of tools to be able to manipulate their genomes. So we can easily cause disruptions in some of the similar genes that zebrafish have to humans. And so in a lot of human diseases, we've traced back some of these diseases to particular genes. And if we then translate this to zebrafish, we can basically cause mutations, the same types of mutations that are in human diseases, in zebrafish and recapitulate some of these diseases. So for example, melanoma is a very widespread type of cancer in humans. It's a cancer of the skin. And there are some particular mutations known to cause melanoma. Well, if we cause those same mutations in the same genes in zebrafish, we can actually cause the zebrafish to develop melanomas in their skin as well. So not only do zebrafish have a lot of similar organs, but it seems like the way that their genes are working in their cells is in a way that's very similar to humans. And so why are zebrafish so popular compared to mammals? There's a lot of reasons. So zebrafish, for one, when they spawn or mate with each other, they produce hundreds and hundreds of offspring, versus things like mice might only produce, say, 10 pups. And because we can produce so many from individual crosses, we can do what we call high throughput screenings. So we can take these baby zebrafish, for lack of better words, and we can treat them with different drugs, or we can manipulate their genes in different ways, and we can see how these things affect a large amount of zebrafish, instead of causing mutations in maybe just a handful of mice. But in zebrafish, we can easily cause these perturbations in hundreds of animals and see how that's affecting the organism. And we can get a better look, a more robust look at how these perturbations are affecting the animals. Additionally, zebrafish are a popular model organism because they develop externally. So the zebrafish lay eggs that are fertilized outside of the body, versus mammals like primates or mice, they have their embryos develop in utero, so within the mother of the animals. So because the zebrafish larvae or embryos are developing externally, this makes it easy for us researchers to actually observe them during development. So we can watch cells move around, we can see the organs develop, and it also makes it very easy to manipulate their genes.